Hello guys, good morning. You're welcome to my YouTube channel, The Explicit Tutors. My name is Tutor Joseph. I teach the various science courses, and the name of my tutorial on my YouTube channel is The Explicit Tutors. Please let me give you a piece of advice before the lecture commences fully, or before I start this video fully, or before you start playing the video. I would advise you, please, you get a pen, a note, and a sachet water beside you, or even chilled mineral, so that as you are listening to everything I'll be saying, it will be entering into your cerebrum. Okay, so please, we are still treating nervous system in biology, and I shall be taking types. I shall be taking the types of neuroglia, all right, in the central nervous system. Now, I told us that there are 4 types of neuroglia. In our previous class, the video has been uploaded already. I told us that there are 6 types of neuroglias. 4 are found in the central nervous system and 2 are found in the peripheral nervous system. Now, what are these 4 types of neuroglia? Number one is the astrocytes. All right, the astrocytes are the most abundant and versatile cell in the central nervous system. They are the most abundant and versatile cell in the neurons, or in the central nervous system, and they have the shape of what? Sea anemones. All right, please take note of that. They ask you which of these type of astrocyte has the shape of what? A sea anemone. Please take note. The astrocytes have the shape of what? A sea anemone, and they are the most abundant and versatile cell in the neuron. Now, what are the functions of these astrocytes? One, they control the chemical environment. They control the chemical environment around the neuron. All right, there is a control the chemical environment around the neuron. All right, they're also important in mopping leaked potassium ions. They mop or cleanse leaked potassium ion, and recapturing and recycling of released neurotransmitters. All right, which means that they control the chemical environment around the neuron, and that is one. And they mop leaked potassium ions, recapturing and recycling released neurotransmitters. You know that neurotransmitters are neurons that have the ability to transmit impulses in the form of what? Electrical and chemical what? Signal. That is number one. Then number two, all right, they make exchanges, all right, between neurons and the brain. All right, please take note of that. They make exchanges between the neurons and the brain, like they exchange materials between the neurons and what? The brain. Secondly, they determine the permeability. All right, the permeability of the capillary. All right. They determine the permeability of the capillary. You know that the capillary is found between the arteries and the veins. They have the ability to carry both oxygenated and deoxygenated what? Blood. So please take note of that. Okay, they also help in the formation of synapse. Alright, they also help in the formation of what? Synapse. Please take note of that. And they also protect or guide young migrating neurons, all right, which means that young neurons, the movement of young neurons are protected by the astrocytes, and the chemical environment around the neuron is controlled by the astrocytes. All right, the mapping of leak potassium ions, recapturing and recycling of released neurotransmitter are under the function of what? The astrocytes. Number 3, they respond to near what? Neurons. They respond to nearby neurons, thereby releasing. When they respond to nearby neurons, they release what? Neurotransmitters. All right, which means that the response to neurons by these neuroglia stimulates the release of what? Neurotransmitters. That is number 3. Then number 4, they have this they influence or stimulates neuronal function, or functioning rather. All right, they influence or stimulates neuronal functioning, and they participate in information processing in the brain and what? Spinal cord, which means that the processing of raw data, all right, into information, or the processing of impulses in the brain and the spinal cord is under the influence of the astrocytes. Please, these are the functions of the astrocytes. I said that they have the shape of the sea anemones, and they are the most abundant and versatile cell in the neurons. So please take note of that. Now the second one I gave in a previous class, of course I listed the 4 types of neuroglia in the central nervous system. The first one is the astrocytes, and the second one is the microglia. Please, I'll rub off this, okay? All right, number 2 types of neuroglia in the central nervous system is the microglia. All right, these microglia are small ovoid cells. All right, they are small ovoid cells, and they have long thorny processes. All right, they have long thorny processes. Now the word thorny is adjective. Now thorn is noun. Now this thorn, the one they call shukushuku, the one they call shukushuku, this RN, this one they call shukushuku is called what? Thorns. Right, which means that it has long projections that look like what? Thorns. All right, that is for that. Now, what are the functions of the microglia? 1, please let me say this. In the neurons, where there are microorganisms, anywhere around the neurons where there are microorganisms or dead neurons, which means that whenever they sense microorganisms or dead neurons in the body, what will they do? They'll bring themselves together in the form of uh, macrophage, and phago phagocy phagocytosis phagocytosis. Yes. Now, when they form when they sense microorganisms and dead neurons around the body, they bring themselves together in the form of what? Macrophage, and phagocytizing or phagocytizing. Sorry, phagocytizing. These microorganisms are what? Dead neurons. Now, these dead neurons can also be called what? Debris neurons. Okay, that is for that. Then two, they are in close contact to nearby neurons. All right, they are in close contact to what? Nearby neurons. What do? They monitor the health of these what? Neurons. Okay, and please, they monitor I said they're in close contact, they're in close touch with what? Nearby neuron, and they monitor the health of the neuron. And whenever they sense injured neurons or similar troubles, what will they do? They will bring themselves together and move towards the injured neurons or any similar trouble that has to do with the neurons. I said they respond to nearby neurons. They also monitor the health of the neurons, and whenever they sense certain injured neurons or similar problem, what would they do? They will bring themselves together and migrate or move towards such injured neurons. These are the functions of the microglia. Please take note of that. Now, the third one is, I'll take the third one, which is the oligodendrocyte. The third one is what? Oligodendrocyte. All right. These oligodendrocytes, what they do is they line the thicker nerve fibers. All right. They line the thicker nerve fibers in the central nervous system, thereby resulting to the production or formation of insulating membrane called myelin sheath. All right. I'll recap. I said that the oligodendrocytes, they line the thicker nerve fibers which are present in the central nervous system, thereby resulting to the formation or to the production of a membrane, which is actually what? Insulative in function, right? It's an insulating membrane called what? Myelin sheath. That is the function of the oligodendrocyte. Now, I said something in our previous class. I said the function of the oligodendrocyte in the central nervous system is the same as the function of the Schwann cells in the peripheral nervous system. Then I also said that the function of the astrocytes in the central nervous system is the same as the function of the satellite cells in the peripheral nervous system. Now, the fourth one, which is the last type of neuroglia in the central nervous system, is the ependymal cells. All right, ependymal cells. Now, these ependymal cells are otherwise called wrapping gametes. That's otherwise called what? Wrapping gametes. Now these ependymal cells, most of them some of them have cilia. Okay. Some have cilia. In some organisms like Paramecium caudatum, the cilia is used for what? Locomotion. Then in some organisms like the sponges, they use the cilia for feeding. By the beating action of the cilia, propels food nutrient to the mouth of the organism, which means that as the sponges beat the cilia, food materials are brought to the mouth. All right, that why this cilia is also used for what? Locomotion in Paramecium caudatum. Now please, these epidermal cells, they range from some are squamous in form, while some are columnar in form. Some are what? Squamous, while some are what? Columnar. They'll ask you which of this type of neuroglia in the central nervous system ranges from squamous to columnar form. It is what? The epidermal what? Cells. And they have what? Cilia. By the beating action of the cilia, since some have cilia, by the beating action of the cilia, it circulates the cerebrospinal fluid. All right, it circulates the cerebrospinal fluid, which have the ability to cushion the brain and what? Spinal cord. You know that the brain has three membranes: the dura mater, the arachnoid mater, and the pia mater. The collection of these three membranes which surround the brain is called meninges. All right. Now, the dura mater, there's a space between the dura mater and the arachnoid mater, and that space is separated, or the dura mater is separated from the arachnoid mater by the subdural space. Then the arachnoid mater is separated from the pia mater by the subarachnoid space. Therefore, the space between the arachnoid mater and the pia mater is, there is a fluid. There's a fluid between, there's a fluid in the space that is between the arachnoid mater and the pia mater called the cerebrospinal fluid. All right. Now, of course, you know that the brain in the skull is not static. The brain is found floating on a fluid. That fluid is called what? The cerebrospinal fluid. And this fluid is found occupying the space that is between the arachnoid mater and the pia mater. I said that the arachnoid mater is separated from the pia mater by the subarachnoid what? Space. Alright, so please take note of that. It is very, very important. Now, I said by the beating action of this cilia, propels or circulates this cerebrospinal fluid, thereby cushioning the brain and the spinal cord. Cushioning, that is, it prevents it from what? Running out. That is the word. Then, secondly, these same epidermal cells, they are found surrounding the ventricles of the brains, or the central cavity of the brain and what? Spinal cord. Okay, now these same epidermal cells, they have the ability to form membranes that are fairly permeable. All right, that are fairly what? That are fairly permeable. Now, this word fairly permeable is that to an extent it permits the entering of materials. The permeability of a material means the ability of that material to be selective to be selective in function. That is selective permeable membrane. It is not all materials it allows inside. There are some materials that cannot enter into the cell, such as materials that are completely solid. It cannot pass through a permeable what? Membrane. Okay, now which means the epidermal cells, they form membranes that are fairly what? Permeable. And the cavity they formed is made up of this fluid, this cerebrospinal fluid. And this fluid, I said, helps to bathe the cells that are found in the central nervous system. Those are the two functions of the epidermal cells. I recap. One, by the beating action of the cilia, circulate the cerebrospinal fluid, thereby cushioning the brain and the spinal cord. And secondly, they form a fairly permeable membrane, and this membrane is made up of cavities that are made up of cerebrospinal fluid, and this fluid has the ability to bathe the cells that are found in the central nervous system. So please, this is all about the 4 types of the neuroglia found in the central nervous system. Now, in our previous class, I said we have 6 types of neuroglia. 4 of it are inside the central nervous system and 2 are in the peripheral nervous system. The 4 are 1, I said astrocytes, 2 microglia, 3 oligodendrocytes, and 4 epidermal cells. Another word for the epidermal cells are called what? Rapid gametes, and they range from squamous form to columnar form, and some of them have cilia which propel or which circulates the cerebrospinal fluid, thereby cushioning the brain and the spinal cord. Then the 2 that are found in the peripheral nervous system are the satellite cells and the Schwann cells. I told that the satellite cells, they form myelin sheath, and the satellite cells, they protect the cell body of the peripheral nervous system. The function the astrocyte executes are the same as the one the satellite cells carry out in the human body. Okay, now I said that the function of the Schwann cells, which they form myelin sheath, and they also found they form myelin sheath, and their function is the same as the function of the oligodendrocyte in the neuron, which means that if the oligodendrocyte line the nerve fibers, that is thicker length fibers, forming myelin sheath, it means that the Schwann cells also form myelin sheath, and the Schwann cells also line larger nerve fiber. Okay, please, not thicker now, larger nerve fiber, which means larger nerve fiber is to the Schwann cell, while thicker nerve fiber is to the oligodendrocyte. Please keep subscribing to my YouTube channel. Do have a wonderful day.